Here we have my LEGO strand of DNA. DNA is a double helix, which means it's made of two separate strands. Each strand is made up of essential parts called nucleotides. Each nucleotide consists of three main parts: a phosphate, a sugar called deoxyribose, and a base, which varies for each nucleotide. As you can see, my DNA strand is made up of 9 nucleotides. In DNA, there are 4 bases: thymine, which is green; adenine, blue; cytosine, red; and guanine, brown. Notice how each base pairs with another base in the opposite strand. These are called base pairs. Each base pairs with a complementary base, forming a weak hydrogen bond. Generally, the rule of thumb is thymine pairs with adenine and cytosine pairs with guanine. Think of it as green pairs with blue and red pairs with brown. Now we'll begin the process of transcription from DNA to mRNA. First, an enzyme called DNA helicase will unzip the DNA. Once the DNA has been unzipped, a complementary strand of mRNA will form and bond with the DNA. Notice that the sugar in the mRNA is black rather than yellow. This is because in mRNA the sugar is ribose rather than deoxyribose. The mRNA strand is exactly complementary to the existing DNA strand. The only difference is, however, the base thymine has been replaced with the base uracil, which pairs with adenine. Now, in RNA, uracil pairs with adenine, or blue pairs with light blue. Once the mRNA strand is completed, it will leave the nucleus and head towards the cytoplasm, where it will then find a ribosome to begin the process of translation. Now we will begin the process of translation from mRNA to amino acid. This is a ribosome, where the process of translation will occur. Translation begins when 3 nucleotides of the mRNA strand, or 1 codon, enters the ribosome. A codon is made up of 3 nucleotides that together codes for a corresponding amino acid. This first nucleotide, AUG, is a start codon. That means that the process of translation can begin. This is a tRNA. It contains a complementary anticodon to the codon on the RNA. It also contains 1 amino acid, which is different for each anticodon. Next, now that the tRNA is bonded with the RNA, the ribosome can move on down the line to the next codon. Another tRNA that is complementary will join. Now, as this occurs, dehydration synthesis will happen, and a peptide bond will form between the 2 amino acids. The first tRNA will leave, and now the ribosome can continue on. The last codon is called a stop codon. A stop codon's corresponding tRNA does not have an amino acid. Instead, the process of translation ends and the amino acid chain breaks off, forming a completed polypeptide chain, or protein. This protein represents 1 trait in your body. Translation has successfully occurred. The mRNA has successfully been translated from codon language into protein language.